Hello viewers, this is Ramakrishnan, Assistant Professor from Department of Biochemistry, St.Joseph's College, Cuddalore. In today's program, I am going to talk about a type of electrophoresis system called SDS page. It is sodium dodecyl sulfate polyacrylamide gel electrophoresis. SDS page is the most widely used technique used for separating the protein in the denatured form. It plays vital role in molecular biology, genetic engineering, nanotechnology, etc., including forensic science. This setup usually follows the separation of proteins based on the size, shape, charge of the molecule. Proteins migrate towards the negatively charged anode inside the polyacrylamide gel under denaturing conditions. Here, protein separation should be denatured with an external reagent called sodium dodecyl sulfate. It is an anionic detergent. So, the denatured protein will migrate towards the electrode. So, separating medium, what we are going to use in this technique is called polyacrylamide gel. This gel provides a porous material that is based on the molecular weight. Separation of the particle will be achieved in the porous material called polyacrylamide gel. When we think about talk about the impact of SDS page electrophoresis system, it should include the buffer tank, lid with the power cables, electrode assembly, cell buffer, then casting stand, casting frame, comb, spacer arm, etc. The thickness of the gel will be decided based on the placing the spacer arm. So, a polyacrylamide gel will be prepared by the polymerization of acrylamide. So, the chemicals required for making these polymers are acrylamide, it is a monomer, and a cross-linking unit called bisacrylamide. Then ammonium persulfate, which act as a free radical inducer for the polymerization. Tetraethylene methylenediamine is a catalyst for this reaction. So, apart from that, water is the medium used for making the solution. So, this is the polymerized gel. So, polymerization is achieved by free radical induced catalysis. In this case, acrylamide group is CH2 double bond, CONH2 amide group, then acrylic compound, so called acrylamide. This acrylamide monomer will be polymerized and forms a polyacrylamide gel. This polyacrylamide gel polymerization will be achieved by the free radical mechanism. Free radicals are generated by ammonium persulfate, presence of ammonium persulfate, and a catalyst tetraethylene methylenediamine. This polyacrylamide gel, it will be in a sheet-like arrangement. But for the separation of proteins, we need a cross-linked gel, just like a fishing net. That cross-linking in the gel will be made by using another cross-linking agent is called N,N-methylene bisacrylamide. So that means two acrylamide units joined through methylene bridge. See this image, CH2CH double bond, CONH, CH2, NHCO, CH double bond, CH2. So two acrylamide units joined through methylene bridge, so called N,N-methylene bisacrylamide. The role of this chemical is it will make a cross link so that we can filter the molecule based on the molecular weight. So, by increasing the concentration of these two, that is a bisacrylamide and N,N-methylene bisacrylamide, we can change the porous size of the gel. Suppose when we want to separate a molecule which is having high molecular weight, we have to use very low, that is large porous size. That means low concentration of the substances. So, how shall we make this concentration proportionate? That will be explained in the following chart, that is image. So, in the polyacrylamide gel preparation, we are using 7% to 15%. Usually a molecule which is having the unit of 50 to 500 kilodaltons, suitable gel percentage is 7%. Then 15% 3 kilodalton to 100 kilodalton. See that when we increase the percentage of the gel, separation will be low molecule. So that means 15% will provide very small porous size. 7% will give a very large porous size. By changing the concentration of the acrylamide in this gel, we will separate the molecules according to their molecular weight. So in the gel electrophoresis, polyacrylamide gel electrophoresis, we are using the gel in two parts. This is the polymerized one. First part is called the stacking gel. The second part is called separating gel. It's based on the concentration. So how shall we make these two concentrations in the gel? So this is the combination. There is a proportionate composition for making the stacking gel and separating gel. So 0.5 molar tris HCl. Buffer is the most important one for maintaining the pH as well as for easy migration by generating the potential difference. In this stacking gel, we are using 0.5 molar tris HCl. Then sodium dodecyl sulfate 0.05 ml. Acrylamide, bisacrylamide 0.67 ml, that is 30%. Then ammonium persulfate 0.05 ml. Similarly, for separating gel, 10 ml proportionate concentration is 0.5 molar tris HCl to 0.6 ml. SDS 0.1 ml, 10% weight per volume. Then acrylamide, bisacrylamide 3.4 ml. So total volume will be 10 and this is 5 ml. Once these combinations are prepared in the presence of the APS and timide, so during this preparation, ammonium persulfate and timide has to be added at the end of the process. That means when we added these two, polymerization takes place. So we should be very careful. After completing all the preparations, we have to add timide, tetraethylene methyl. So there is a catalyst. So we have to add at the end. Once these catalysts and free radical inducers are all added, once the polymerization takes place, then the gel will be formed. In order to make a sample loading well comb, we have to use comb for making this sample loading well. So after polymerization, the gel will be costed in the setup. The setup consists of the upper tank buffer reservoir, lower tank buffer reservoir. The two buffer tanks are all attached through the separating gel, that is a stacking and separating gel, that is called a polyacrylamide gel. Then sample to be separated has to be loaded in this one. Before to load the sample, we have to process the sample. So how shall we process the sample? Since we are following the denatured form of protein during separation, we have to process the sample by using the reagents SDS. So this SDS is a sodium dodecyl sulfate. It is an anionic detergent. It will bind with the proteins, and therefore entire charge of the protein turns into negative charge by denaturing the protein. Look at this image. First before denaturation and after denaturation. The protein is completely merged into a primary structure with negatively charged ions. That means the entire molecule will be negative in charge. So this is the structure of SDS, sodium dodecyl sulfate, so anionic detergents. And apart from that, the sample will also be treated with the performic acid or beta-mercaptoethylamine. These two agents will denature the disulfide linkage present in the protein and makes it into a linear form. So that is why it is a denatured protein separation system. Once the proteins are denatured, then it will be placed into the sample loading well with the help of a tracking dye, bromophenol blue. That tracking dye will help us to find the reaching point. Okay. So at the same time, we are going to load a molecular marker, molecular ladder for proteins. So once the samples are loaded, saturation points are all completed, applied electricity will start the movement of proteins. So upper buffer reservoir is a cathode and lower buffer reservoir is an anode. Since during the process of denaturation all the proteins are all converted into a negatively charged one, so the protein separation will be a unidirectional, that is only towards cathode direction. So proteins will migrate from top to the bottom during electrophoresis. So this is the gel plate during the process of separation. The gel, stacking gel, first part stacking gel, this will helps for aligning the protein before separation. Once the proteins reaches from that is reached and crossed the stacking gel, it reaches to the running gel. So based on the molecular weight, separation of proteins will be achieved. The molecule which is having high molecular weight, it will be on the topmost bottom, its topmost layer. Then according to the linear, that is low molecular weight, so smaller proteins will migrate in a faster manner than the this one. So once the process is achieved, so how shall we came to know the process is over? So we will see the movement of the blue band. So at the end we have to switch off the system. So during this process we have to maintain proper supply of electricity, buffer, etc. Then after the process over, remove the gel cassette that consists of glass plate, spacer, everything. Remove the glass plate and carry the staining process. So after staining we will find the protein bands like this. So first one, here for an example they have mentioned a 2-well gel. First well represents the protein marker one. The second one is the protein what we have used for separation. The marker protein, see that myosin having very high molecular weight, which will be on the topmost layer, and the lysozyme just 14,400 Dalton, that will be in the least molecular weight and migrated to the bottom. So according to the molecular weight we will see the layering of proteins in the gel. So it is called a molecular weight ladder, just like a ladder arrangement. Now the sample well 2 contains the sample what we are going to or what we have used for separation. So a band which is observed in the region close to 31,000 kilodalton, and in between the 45 and 31. So with the help of a gel documentation system, that is computerized system, that will exactly pinpoint the molecular weight by making the graph, so that we will come to a conclusion that the isolated protein is having the specific molecular weight. For the structural study and the molecular findings, this technique is broadly used. In this case, what happens while treating the sample with the SDS compound? So when you take a protein as such, see, look at this image, there are two proteins in this. So one is A, B complex, another one is C. First protein is a protein made by two subunits, A chain and B chain. The A chain and B chains are linked through disulfide linkage, whereas the C protein is a single polypeptide chain protein. When we carry the SDS based denaturation, the two chains A and B of the protein 1 gets broken into two different protein chains, that is A chain plus B chain. A B chain turned to A chain and B chain, A chain plus B chain. When you apply in the electric field, that protein turned into two different bands, that is A band and B band. B band and A band according to the molecular weight. In this picture, B band is having high molecular weight, it will be on the top. See, from the image itself you will understand. A is linear polypeptide chain with the minimum and B is maximum. So maximum will be on the top and minimum will be on the bottom. So this is the way we are separating. When you carry the same experiment without using SDS based beta mercaptoethanol or performic acid, then that electrophoresis setup is called polyacrylamide gel or native gel. That means the protein separation will be in the native state. So for example, when we take A and B, A plus AB protein, it will be in a single band. Rather than the two bands, it will be a single band in the case of native page. That is the major difference in between the page and SDS page. The one and only difference is sample processing. In the case of polyacrylamide gel, we are not using the sample processing. That means absence of SDS, then beta-mercaptan, etc. In the page, we are not using this one. In the SDS page, we are using, that's all. So applications of SDS page. When we look at the major applications of this one, it is generally used for finding and confirmation of various viral infections such as AIDS. It's a confirmation test. Then determining the purity of protein sample. Suppose a protein is isolated from a sample, biological sample, whether it is pure or not, we will conclude by using this electrophoresis. Then determining the molecular weight of a protein by placing in the well and comparing with a gel ladder, we will determine the molecular weight of the protein. Next, identifying the disulfide bond between the proteins. Since we are using the denaturation system where the disulfide bond is present, suppose a protein is made by two chains, A chain and B chain, where the disulfide bond is present, positioned in between the 5 and 6 of amino acid, or 7, 8 like this, we will identifying the position of the disulfide bond in the protein. Then quantifying the protein, amount of protein present in the sample will also be studied. And finally, the latest technique called blotting techniques. That is also once again confirming the diseases, which most important one for in the clinical findings and the confirmation studies that follows SDS page acrylamide. Thank you.